Guys, we are very briefly going to go over the history and composition of blood. These are two very important things that you have to know and have to understand before we can get into the more complex things. So you need to remember the difference between class and individual evidence. Blood typing is class evidence because it can narrow it down to anyone that has AB positive blood, or it can eliminate people based on that, but it cannot tell me exactly who left that evidence. Whereas DNA profiling is individual evidence because it can point me to exactly that person or individual. And then blood spatter helps to recreate the crime scene, and we're going to work a lot with that in our labs during this unit. A little bit of the history. Way back when Egyptians used bloodletting or releasing blood to cure a lot of diseases is what they thought. Then back in 500 BC, the Greeks distinguished between arteries and veins, so they understood that they had two different jobs. In 1659, Leeuwenhoek, which is a name that should be familiar from biology and potentially anatomy as well, he viewed blood cells with a microscope. 1795 was the first blood transfusion. 1901, we had the three blood types discovered by someone named Landsteiner. And those three blood types, or blood categories, I suppose, are A, B, and O. Then in 1902, De Castello discovered the AB blood type. 1922, the blood donor service was established. 1935, the Mayo Clinic. They then developed a method to store the blood for transfusions. 1959, the first AIDS case was recorded in the Belgian Congo. 1984, this guy over to the left identified the virus causing AIDS. And then from 87 to 2002, the development of blood screening tests were used or started to become used for testing for infectious diseases. So as far as what blood truly is, so what makes up blood, 45% of blood is cells, and those cells can then be broken down a little bit more into red blood cells, aka erythrocytes, white blood cells, or leukocytes, and platelets, which are your clotting factors. The liquid portion of the blood is something called plasma, and you can read there the big part of what plasma is made of is 90% water, and then it includes all of the other things listed there. So if we break these down even more, erythrocytes, they are important because they carry gases, and that's probably what you know red blood cells for. They are produced in the bone marrow. They do not have any internal organelles, so they have no nucleus. And then hemoglobin, you probably already have heard this word as well, that contains iron, that contains a protein to bind to oxygen. This is what gives blood the red color that it has. White blood cells are also called leukocytes. This is a good chunk of your immune system, helps to fight disease and foreign invaders. Also produced in the bone marrow, and it is a true cell, and it has a nucleus. Platelets are tiny, tiny cell fragments that help in blood clotting. So when you cut yourself and it starts to scab over all of that, that is platelets going to work. Then this test is used if there is thought to be blood at the crime scene. It's called the Kastle-Meyer test. Let's talk a little bit about presumptive versus confirmatory tests first of all. Presumptive is exactly what it sounds like. It's not concrete. If you presume something, you are basically making a hypothesis or making a guess. And what this test will do, it can say, hey, a sample is not a certain substance, so it can eliminate an option, or it can say a sample probably is a substance. Once they get to that route, they will probably run a confirmatory test, and that says for sure what the substance is. So the different steps to this, you would wet a cotton swab with distilled water, so that's basically purified water, and rub on the stain that you think potentially is blood. And then you see the different steps where the hemoglobin is exposed, phenolphthalein turns pink when oxidized or exposed to oxygen, and then hydrogen peroxide will react with hemoglobin. So basically what will happen at the end of this, and you're going to watch a video over this, if blood is present, the swab will turn pink. Okay, so blood is present, pink. Blood is not present, it'll be white. And you can see in the picture right there, the positive is on the left and the negative is on the right. Sometimes non-blood substances can yield a positive test. Some vegetables and then some different salts can do that. So why do we do this? If we have all sorts of DNA profiling, if we have all sorts of different tests that can be used to individualize this, why do we use it? Blood typing is useful because it's less expensive and it's a lot quicker than DNA profiling. It can be used to link or exclude a suspect, but it can't prove guilt because it's not an individual evidence. Okay. So the discovery of the different blood types is credited to this guy. And what he found is that blood from one person didn't always mix freely with another person. And the clumping that resulted from this resulted in death. So this is due to some different surface proteins, which I'm going to go over with you in class. You have A and B proteins, and then you have something called the RH factor proteins. The RH factor is your positive or negative. If you're AB positive, AB negative. And then we're going to run some antibody reaction tests to identify the different blood types. But you can see the commonality of each of those in the visual aid to the left.